All right, well, welcome back, everybody. I hope a little 15-minute break from the previous session is left feeling you recharged and energized to hear some really exciting talks from our panel of investigators that you can see here. Again, it's really been an exciting few days for me to be able to sort of see what BME is doing and the breadth of interactions that it has. As I've been doing, just again, quickly thought I would plug the DEI Award and Mentorship Award. Congratulations, Chris and Brendan. At the same time, in the news, I'll mention a couple of things. I was the fortunate recipient of the 2021 Society for Biomaterials Technology Innovation and Development Award. This was work for our nanoparticles that's being used for treating autoimmune disease, and specifically I had completed a clinical trial with celiac disease. And so it's definitely fits within this idea of a precision health type of application. Also, we've been fortunate to recruit Aaron Morris to our faculty ranks as well too. And later on this month, you'll be able to actually watch his TED Talk that he received the recognition as a TED Fellow during this past year and had the talk recorded. And unfortunately, he's not able to attend the event in person on stage in Vancouver, but they've worked behind the scenes to do some taping of this. And my understanding is it's actually going to be released in the next couple of weeks. And so stay tuned to be able to watch that. And our final note of in the news with BME is Alberto Figueroa, distinguished faculty member with a joint appointment between engineering and surgery, had been appointed to the American Institute of Medical and Biological Engineers as a fellow. So congratulations to Alberto and Aaron. And I will turn the floor over to Kelly to simply lead off this really exciting panel.
All right, thank you for being here today in our session on precision health. This session will run similarly to the previous sessions in that we'll have a series of faculty talks, 3 faculty talks followed by student mini dissertations. And then at the end, we'll have a panel discussion and a poster session.
Now, my lab, as Kelly mentioned, develops computer models for drug discovery. And today I'm going to talk about some new methods my lab has developed for designing precision therapies for infectious disease. So I think antibiotics are one of the greatest inventions of mankind. You know, before antibiotics were invented, a significant number of people died during childbirth or when they had diseases like pneumonia. And today, due to rising antibiotic resistance, you might have to go back to this scary era where even a simple wound or scratch could become life-threatening. So there are 2 main reasons why there is this antibiotic resistance pandemic. One is the widespread of antibiotic-resistant bacteria. And the second one is that they have, there's a shortage of new antibiotics that are entering the market. So as you can see from this graph here, around the 50s and 60s, we invented a lot of antibiotics. But then now in the last couple of decades, there's hardly been any new antibiotics that have been introduced to the market, mainly because we have sort of exhausted the natural source of antibiotics. And there's hardly any new mechanism or compounds that have been discovered recently. So these 2 combination of spread of drug-resistant pathogens and the lack of new antibiotics is what's driving this pandemic. And another main reason behind this is that currently there's a lack of precision in which the way we treat infections. For example, we don't take into account the properties of the patient or the genotype of the pathogen. Similarly, the way we discover antibiotics also has to change to a more precise approach where we take into account give specific drugs based on the pathogen's properties of the host immune system, and the metabolic environment of the infection site. So today I'm going to talk about how my lab is developing methods to take into account this complexity. So now overall, the computational methods our lab develops sort of is a hybrid between traditional engineering models and machine learning models. So traditional models like metabolic modeling, pharmacokinetics, agent-based models that are really mechanistic. They can give you a lot of insights on the underlying biology, but the problem is that they are not accurate enough to be used in the clinic. In contrast, machine learning methods are really accurate. They have very high precision and recall, but they're usually black boxes. You don't really know how they work, and it's hard to trust these models because they are so opaque. And so what my lab is doing is trying to leverage or combine these mechanistic models with machine learning models so that we can still get high accuracy, but also underlying mechanism that you can get from engineering-based methods. So today I'm going to talk about, like 4 key challenges in drug discovery for antibiotic resistance using the methods my lab has developed. So I'll initially talk about how we can use AI and machine learning to screen through large number of drugs and drug combinations. And then I'll talk about how we can apply these methods for systems like pathogens like Staph aureus, shown here, where you don't have a lot of omics data. And then lastly, I'll talk about how we can better simulate the in vivo environment, its metabolic properties, its immune properties, and take that into account while designing drugs. So today I'm primarily going to talk about how we're going to design combination therapies. I think that's like the major new frontier for infection and even in cancer. The reason being that combinations are really good at reducing the rise of resistance. And another advantage of combinations is that you can combine them and get really non-intuitive effects. Like you can get very high potency or, like extended spectrum where you can kill multiple pathogens at the same time. And you can also repurpose drugs that have low potency, but combining them together, you get, like really novel combinations. So I'll talk about examples of these during my talk. So for the first challenge, we developed an approach called Indigo. So what Indigo does is that it predicts synergy or antagonism of drug combinations. The way Indigo works is very similar to how, like Amazon's product recommendation or Netflix movie recommendation works. A Netflix algorithm takes in 2 inputs. One is your prior movie watching history, and then it has an underlying database of movie properties. Like, is a movie a horror movie or a comedy? So knowing these underlying properties, it's able to recommend new movies based on your past movie history. Similarly, Indigo takes into account known examples of drugs that are synergistic, and then it connects that with underlying properties of drugs, and it's able to predict for new drugs if they'll be synergistic or antagonistic. So the drug properties that Indigo uses is what's called as a chemogenomics or drug gene interaction data. And we can get this in multiple ways. One common way is we use transcriptomics profiles of cells treated with different drugs. So you can find, using transcriptomics data, what genes are going up or down after drug treatment. And then you can create a drug gene interaction network from that. Another data set we use is genomics data that tells us information on what genes, when mutated, cause resistance to an antibiotic. So using this for each drug, we can get sort of like a signature of genes that it's known to interact with. So it's sort of similar to the movie properties that I mentioned for Netflix. So knowing these drug properties, the Indigo can then connect these to predict how 2 drugs or 2 or more drugs will interact. Will they be synergistic or antagonistic? So that's the underlying idea behind Indigo. And then we have tested this in a wide range of organisms. So I'm showing data initially for E. coli, where we measured synergy or antagonism between diverse types of antibiotics, shown here, where red is antagonism, blue is synergy, and white means they don't interact. And using this small set of training data between like 20 different drugs, Indigo then connects that to underlying drug properties, shown here on this chemical genetic matrix. And now it can make predictions for all the drugs for which it has chemical genetic data for. So in this data set, Indigo was able to make more than 50,000 predictions of pairwise drug combinations. And then we experimentally tested the strongest synergy and antagonism, which we then confirmed experimentally that Indigo is able to accurately predict drug interactions for novel drugs with even new mechanisms of action. So this is all for E. coli. So next I want to talk about how we can apply the same idea to less studied pathogens. So this is where building sort of semi-mechanistic machine learning models helps, is that since we know the underlying principle behind how Indigo works, we can sort of leverage that to apply it to different contexts. So specifically, indigo works by building these drug-gene interaction networks. And what we discovered was that if we can identify genes used by indigo that are highly conserved between two species, for example, E. coli and Staph aureus here, then what we hypothesized was that drug interactions that depend on conserved genes would also be conserved in a different organism. So if you look at the machine learning algorithm here, for example, if gene 1 and gene 2 are conserved between E. coli and Staph aureus, then the corresponding interaction synergy might also be conserved. Whereas if gene 3 is not conserved, then this antagonism might not be conserved. So by using this data set from E. coli, we could potentially predict interactions in new pathogens. And so then we tested this in Staph aureus, where we measured, once again, drug interactions between different antibiotics. And to our surprise, we found that using just the data from E. coli, we were able to accurately differentiate synergistic drug interactions from those that are antagonistic or non-interacting. So we got this data for free. We're using data from a different organism and making predictions for a clinically relevant pathogen. Another system we apply this is Mycobacterium tuberculosis. Tuberculosis is a deadly pathogen that kills more than 1 million people every year. And then due to drug resistance, new antibiotics are urgently needed. And so we teamed up with David Sherman's lab at University of Washington to apply indigo to this pathogen. So with David Sherman's lab, we also generated new data sets now that we can then expand our indigo model. So this model is actually bigger than the E. coli model that I showed earlier. So using this tuberculosis indigo model, we're able to predict more than 5 million drug combinations of 2 drugs and 3 drug combinations. And we then tested the most promising synergistic ones experimentally, and and it showed great correlation with what was observed experimentally. And one cool combination that we found was involving 2 drugs that are not normally used for treating tuberculosis. So this drug here, chlorpromazine, is actually an antipsychotic drug, and spectromycin is an antibiotic that's used for a different disease. And what we found was that indigo predicted them to be highly synergistic. And you can see from the experimental data here that when spectromycin is given on its own, only at 2x its dose, it kills all bacteria. Chlorpromazine also only kills all bacteria at 1x its dose. But when you combine them, 1/16 of spectromycin and 1/16 of chlorpromazine is able to kill all the bacteria. So it's at least a 10-fold increase in potency. And so now we can repurpose drugs that are used for other diseases for treating tuberculosis. So the last few minutes, I want to talk about how we can better predict the impact of the infection microenvironment, like metabolism and immunity, on these drug combinations. So recent studies have found that the metabolic environment where the pathogen is has a huge impact on how it responds to antibiotics. So certain metabolites produced by your host cells or maybe the microbiome can protect the pathogen from antibiotics. And so we want to take this into account while we discover drugs or design new treatments. And so to take this into account, we developed an updated approach called Magenta, which is similar to indigo, but in that it also takes into account the metabolic environment in which the bacteria is growing in. So if it's encountering glucose or glycerol, so it's taking into account the genes that are needed by the pathogen for growing in glucose or glycerol, and then it makes condition-specific predictions now. Instead of just saying drug A and B are synergistic, it says drug A and B are synergistic when glucose is present, but they might be antagonistic when a different metabolite is present. So now using Magenta, we can make more nuanced predictions depending on the metabolic condition. So we applied this to Acinetobacter baumannii, which is once again a very deadly pathogen common in hospitals. And what we found was that depending on the metabolic condition which the bacteria is grown in, you can see here that the drug interactions change dramatically. Drugs that are antagonistic in LB media, which is a commonly used lab media, become synergistic in glucose or glycerol conditions. You can see that each condition has its own unique profile. And so you need methods like Magenta to screen through various metabolic conditions. So the last thing I want to talk about is how do we capture the infection environment in itself to design better therapies. So for this, now we work with Jennifer Lindeman's lab in chemical engineering and Denise Kirshner's lab in microbiology and immunology to combine indigo with an agent-based model that they have developed for tuberculosis infection called Granson. So the idea is that so far, we've been testing indigo or drug combinations in lab conditions, whereas inside the body, there are a lot of barriers, like biophysical barriers, that could prevent the drugs from reaching the same infection site. And there's tissue pharmacokinetics where some drugs could diffuse in faster or leave the infection site faster. And so we need to take those things into account while we design therapies. And so what their lab has developed is an agent-based model of the of MTB infection, where the pathogen is surrounded by macrophage and other immune cells. And so the drug has to penetrate this ball of cells to actually kill the bacteria. So now we can, using their model, we can take into account how drugs penetrate the infection site. Do they reach the site at the same levels to be synergistic? And so we can make more nuanced predictions. So by doing so, now we can sort of start at the molecular level and go all the way to, like whole body physiological levels, where with indigo, you can predict drug synergy at the cellular space. And with Granson, we can account for the tissue level changes. So using this now, we have predicted outcomes for clinical trials, where each chart I'm showing here is a clinical trial of a drug regimen for tuberculosis. And you can see that by combining indigo with Granson, we're now able to accurately predict clinical trial outcomes. So now we are taking this approach towards personalized medicine, where we are looking at health record data, and you're trying to take into account chest x-rays of patients, attributes of pathogen, like what mutations they have, and then try to predict what the outcome would be, like whether this patient will survive, is that person at risk for reinfection or relapse, so then we can make more personalized decisions for different infections. So just to summarize, I talked about 4 key challenges, talked about how we can screen through a large number of drugs and drug combinations using indigo, how we can apply this for many clinically relevant pathogens using indigo orthology, how we can simulate metabolic environment using magenta, and how we can simulate the infection immune environment using Granson. So with that, I want to thank you all for your attention and thank my collaborators, students, especially Suraj and Chen, who led many of these projects, and funding sources, especially Precision Health, for providing pilot funds for this. And thank you all.
Now we'll get started on a series of student mini dissertations that were pre-recorded, will be played now, and then after that we will have the panel discussion.
Hi, I'm Christina Lee. I am a third-year PhD candidate in Kelly Arnold's lab, and today I'll be talking to you about the vaginal microbiome. So you're probably familiar with the health benefits of the gut microbiome, ranging from improved digestion to even improved cognitive function, but other microbiomes, such as the vaginal microbiome, are also really important for our well-being. However, the vaginal microbiome commonly undergoes shifts from an abundance of good or beneficial bacteria to an overgrowth of bad or potentially pathogenic bacteria, leaving women at higher risk for adverse reproductive outcomes, as well as increased likelihood to get sexually transmitted infections. This dysbiosis, called bacterial vaginosis, is notoriously difficult to treat, with 1 in 2 women failing standard antibiotic therapy. There are many reasons this could be occurring, but one such reason could be that many bacteria are working in concert or antagonistically to regulate the complex vaginal microbiome ecosystem. Thus, we have built mathematical models to encompass these interactions so we can better understand, quantify, and predict the behavior of the vaginal microbiome to interventions such as antibiotics. So far, it's been really helpful in identifying key personalized differences in vaginal microbiome composition that affect efficacy. Surprisingly, higher levels of good bacteria relative to bad bacteria were associated with higher risks of treatment failure. In other words, a less severe dysbiosis was associated with decreased antibiotic efficacy. On further evaluation, our results suggested that good bacteria were sequestering the antibiotic and to an extent that reduced the amount that could act on and inhibit the bad target pathogenic bacteria. Now we are continuing to develop our models so we can test hypotheses that are either intractable or challenging to test clinically to inform the development of things like probiotics, prebiotics, and antibiotic regimens. Thank you.
Hello, my name is Ben. I'm a fourth-year PhD student in the Aguilar Lab, and I'd like to talk today about my project concerning metabolic regulators of muscle stem cells in injury and aging. The loss of muscle mass and function with age, also known as sarcopenia, presents a large financial and clinical burden that is partially driven by losses in the number and function of muscle stem cells that support muscle regeneration. In this project, we study sestrins, a class of stress-inducible proteins with antioxidant functions that have yet to be studied in adult muscle stem cells. In mammals, sestrins 1, 2, and 3 regulate metabolic responses to oxidative stress by physically activating AMPK signaling and indirectly suppressing mTORC1 activity. These combined efforts upregulate autophagy and maintain proteostasis, which ultimately preserves muscle mass and function. We use skeletal muscle sestrin knockout mice models, referred to as SKO, to observe the influence of sestrin loss on muscle stem cell homeostasis. Young, uninjured wild-type and SKO mice exhibited no change in their number of PAX7-positive muscle stem cells, but showed an increase in the fraction of stem cells expressing PS6, the mTORC1 target, and the cell proliferation marker Ki67. Immunofluorescence staining of freshly isolated muscle stem cells revealed decreased quiescence depth and increased oxidative stress in SKO muscle stem cells. Transcriptomic analysis before and after injury showed that while sestrin loss alters basal-level transcriptomes, the regenerative response is unmodified. Moreover, SKO muscle stem cells upregulated genes before injury related to oxidative stress metabolism and cell cycle checkpoints. Collectively, these results show that loss of sestrins 1 and 2 doesn't significantly alter muscle regeneration, but drives differences in metabolic pathways to hyperactivated mTORC1 signaling that contribute to reduced quiescence and premature activation in muscle stem cells. Next, we observed a previous gene expression data set of young and aged muscle stem cells before and after injury and saw reduced sestrin 1 expression in aged muscle stem cells during regeneration. To probe further, we examined muscle for middle-aged SKO mice and observed a loss of muscle stem cells compared to wild-type mice and an increase in the fraction of PS6-positive muscle stem cells. We also saw smaller myofibers in SKO tissues with comparable numbers of centrally nucleated myofibers between conditions. Finally, we examined the regenerative capacity of SKO mice with age through double injury and found comparable numbers of muscle stem cells, but an increase in the PS6-positive fraction. We also saw an increase in the fraction of activated stem cells and committed myogenic progenitors in SKO muscle tissues, along with enhanced collagen deposition and a gain in glycolytic type 2B muscle fibers after injury. Overall, this project reveals sestrins to be important metabolic regulators of the quiescent state in adult muscle stem cells, and further study may advance our understanding of stem cell biology across lifespan.
Hello everyone, I'm Natacha Comandante-Lou from the Falahi Lab. Today my presentation is about investigating the role of AP1 in directing melanoma heterogeneity and resistance. Targeted therapies often show substantial short-term effects, yet fail to achieve full cures. While resistance can result from genetic causes, it can also emerge from non-genetic variability among individual cells. This enables a subpopulation of tumor cells to transiently tolerate the drug and escape therapies. A prototypical example of this is seen in BRAF mutant melanoma treated with BRAF inhibitor vemurafenib. Numerous studies have linked this non-genetic variability to the differences in melanoma differentiation states or to different feedback mechanisms involved in signaling homeostasis. It is not clear how or whether these seemingly distinct mechanisms are connected. Here, our working hypothesis is that at least some aspect of these adaptive mechanisms can converge to a family of transcription factors called AP1. AP1 is relevant because it is directly downstream of the targeted MAPK pathway and has been implicated in the regulation of drug resistance and differentiation in different tissues. Using partial least squares regression modeling, we have shown that AP1 is predictive of baseline melanoma differentiation states at the gene expression level. The AP1 model outperformed most of the random combinations of transcription factors. Integrating multiplexed immunofluorescent imaging and data-driven modeling, we've also found that AP1 is predictive of differentiation states at the single-cell protein level. These analyses provide us a list of AP1s that are associated with each melanoma differentiation state. In the case of drug treatment, we have also found different MAPK inhibitors of different doses can induce variable trajectories of changes in AP1 states at the single cell level. Altogether, this systematic analysis suggests the key role of AP1 transcription factor network in linking diverse melanoma differentiation states and MAPK inhibitor responses. With that, I would like to thank my lab, my committee members, and our funding sources. Thank you.
Hi, my name is Claire Tomaszewski, and I'm a fifth-year PhD student in the Shikhanov lab. We do ovarian tissue engineering because unfortunately, chemo and radiation lead to infertility in women and young girls undergoing cancer treatment. Some women are able to undergo hormone stimulation for egg retrieval, and these eggs can be cryopreserved. But women who cannot delay cancer treatment for hormone stimulation and young girls who have not yet undergone puberty are not eligible for this procedure. However, in vitro follicle culture holds promise as a broad fertility preservation option for these patients. They would simply freeze some of their ovarian tissue prior to getting cancer treatment, and we could later mature follicles in vitro to give them a fertilizable egg. My research focuses on designing biomaterials for culturing these ovarian follicles. Each follicle contains a single egg surrounded by hormone-producing cells, and these follicles depend on complex temporal and spatial cues to mature. These signals come from both the surrounding follicles and stromal cells, as well as the extracellular matrix. So our goal is to recapitulate these cellular and extracellular components for maturing follicles in an artificial ovary. Using a polyethylene glycol matrix, we co-culture follicles with ovarian stromal cells to incorporate cell-cell interactions. To mimic cell matrix interactions, we use protease-sensitive crosslinks so that the follicles can degrade the matrix as they grow. And we've also included ECM binding peptides so that the follicles and stromal cells can rebuild their native ECM within the PEG hydrogel. The retained ECM can then further bind growth factors and cytokines for improved paracrine signaling. While we have designed this material for follicle culture, it could theoretically be expanded to many other cell types. By retaining cell-secreted ECM, the cells themselves are determining the ECM composition and can rebuild their native environment. So in summary, by co-culturing follicles with stromal cells in an ECM sequestering matrix, we have built an artificial ovary which facilitates follicle maturation and yields mature eggs. This is a broad fertility preservation option which would allow cancer survivors to have biologically related children without the risk of reintroducing cancerous cells and without delaying cancer treatment. With that, I would like to thank my lab, collaborators, and funding.
All right. So thank you to the students for those fantastic presentations. Those were really great. And I guess now, I think we're going to move on to our panel discussion, Hope or Hype for Treating Diseases with Precision Medicine. And our panelists are all of our speakers that we just heard, the faculty speakers. And so we will get started now. And I guess I thought I would just start things off by talking a little bit about precision medicine. And I think, you know, precision medicine, it's so exciting, this idea that you can take personalized measurements from people and then use them to address disease heterogeneity in individuals and in theory, make tailor-made treatments. And so I was just, and I think this theme has just emerged recently. Yeah, if we look at the history of the Precision Health Initiative at Michigan, it only started in 2017. And so the first thing I wanted to see if maybe the speakers could comment on is why now? You know, why are we at a place right now where we can do precision health? And I guess especially I'd be interested to hear, you know, in the context of your own research and where, you know, the tools that are available. So I guess maybe Sriram, could you give us do you have any thoughts there?
Yeah, sure thing. Yeah, so, well, I guess it's twofold. One is obviously, you know, people want to do, you know, if they want to do personalized medicine before, you know, they didn't have the data or the technology to do it, you know, with RNA personalized sequencing and, you know, with new algorithms coming out. I think it's a combination of both of these that, you know, it's driving precision medicine. So I guess, I mean, talking about even my own research, you know, if you want to do more precision therapies for infection, it would have been hard earlier. Like we didn't know, like there is no point of care measurements that could be made on the patient or pathogen, and people didn't have these systematic health records that could say, you know, like this person died or this person, survived this treatment. So I think it's a synergy of both those things that's accelerating it. And probably once we have better data sets, I think we'll eventually see that in the clinic soon. But so right now we're still, exploring research, trying to see if it's possible. But I think maybe in a few years we'll actually see that in action.
Maybe I can add on to that from my perspective on cancer immunotherapy. So there has been a lot of work prior to this on the personalized immunotherapy approach. And now we are seeing the fruits of the hard labor. First, we have the high throughput screening method that allow for identification of patient-specific mutanomes. And also the in silico prediction tools are available to predict which of the mutated epitopes are presented on tumor cells. And the ways to generate patient-specific vaccines or patient-specific CAR T cell therapies are being available, largely driven by academic centers, but now there are biotechs and companies that are investing in this. In particular, for example, personalized cancer vaccine is spearheaded by BioNTech, the makers of the Pfizer vaccine for COVID-19. So rather than using mRNA for COVID-19, they are encoding patient-specific antigens using mRNA, and they are the ones who did a phase 1 trial first, and they're moving into like a glioblastoma and other cancer patients. So it's a really exciting time to be in this, but there are many challenges. Number one will be the precision, like how well you can predict the right antigens, as well as the cost. Because it's very costly to go through all these personalized therapeutics and production of like drug products in a GMP setting. So that's the remaining challenges.
Just to add to that, what Sriram and James said, so I think, they both addressed about the technological advancements, I think, which have been made, which allows us to do it now, specifically like related to single cell RNA-seq, and also in terms of our metabolic data is like coming up from the metabolic imaging, which can resolve the spatial heterogeneity or the spatial aspect related to the omics. So that has been I think, which is the most exciting thing, that data coming and will help us, and I think address this issue of the individualized therapy.
Yeah, it's interesting. It sounds like in all three of your research areas that it's like these cost-effective technologies that let us make high throughput measurements in a personalized way. And now that we have these, we can move forward. And I think, James, I think you touched on this, but I think this sort of now, like, so what are the biggest challenges? And James, you mentioned this too in your research, but I was curious if Deepak or Sriram, if you have anything to add in terms of, in your research areas, what are the biggest challenges now that we have the measurements? What are the biggest challenges in moving forward with the therapies, the personalized therapies?
I can maybe start. So I think the challenges again are, although I said that technological innovations are there, but still it's almost like an Heisenberg uncertainty principle. Like the more now you can go to the one cell, but coming back to the means again, like the speed and the position and the velocity, same cannot be measured. So same thing here. You can find the individual cell, but how do you connect it to the whole? I think will be the big issue, I personally feel. So that's like reconstruction, going from back to and building up. So starting from one cell to the building up will be a big challenge. And I feel how the tissues are formed and how the diseases are formed. So I think in that respect, how to utilize this technology, this mammoth data, and which is like which again will be very. I think it will be a big challenge for both for generating the people who are generating the data and also on the modeling side who are integrating the data.
Yeah, and I guess another thing I would add from what Deepak said is, I think for many of these modeling or algorithms, they sometimes work really well in one setting, but expanding them to multiple settings is extremely challenging. Might work in Michigan, but not in Texas, so they seem to learn patterns that are unique to a data set, and I think that's a huge challenge. We can get good accuracy in a very controlled environment, but making this a mass like treatment is going to be, I think, a huge challenge. So, and I think there's going to be, might work in one person, but not the other. And then explaining why it worked for one person and why it did not work for another person is also a huge challenge. And I think especially for clinicians, if they're explaining to a patient that, hey, this algorithm said, give this drug, they won't really know why it said so. And I think that once again, the trust issue is there and the scalability. I think those are big challenges that we need to focus on next.
Yeah, thanks. I think those are really interesting thoughts. I think, Shri, to your point, the mechanism, like how do we get from a signature for a person to understanding the mechanisms of disease? And maybe that's even like perhaps related to what Deepak was saying about how do we translate personalized measurements across physiological scales? We might have a personalized understanding at the molecular protein level, but how do we translate that to function and disease state? And so, yeah, those are great thoughts. I was curious if you guys had any thoughts on, as biomedical engineers of the biomedical engineering department at the University of Michigan, what are like some unique contributions or a unique perspective we have on precision medicine, and how do you think we can contribute as biomedical engineers?
I guess there are multiple ways. So, I guess from the symposium itself, I learned a lot, like starting with imaging. Biomedical engineers are really good at, like creating technologies for imaging, but also, I guess, developing algorithms for analyzing them. So I think BME could be involved in every aspect, from data generation, developing technologies for sequencing, once again, analyzing the sequencing data, metabolic measurements, vaccines. So I feel like BME, biomedical engineers can be there from every step of the process, from imaging for diagnostics, like point of care measurements. And so I think every aspect of precision medicine could be influenced by biomedical engineers.
Yeah, I mean, just to add to Sriram, I think that tissue engineering is which is our core. I think tissue engineering, imaging, which are our core, and also the understanding the cellular scale, the quantitative aspect, which many of us specialize. So that I think is the core strength, and which we can use, which is I think the unique strength of compared to the other engineering departments or even the life sciences other departments. I think we can strongly, once we have integrated tissue engineering, imaging, and the quantitative omics, if we integrate, I think we are well suited to integrate it as researchers in our department.
And just to add a little bit, I think biomedical engineers are excellent at running basic studies as well as translational studies, and they are very excellent in communicating basic science as well as complicated theoretical framework to the general public. And I say that because when you think about running clinical trials with a personalized precision medicine, you have to convince FDA that this is needed and this how to run clinical trials as well. And they're not as well versed in how to design clinical studies and how to basically tailor drugs to individual patients, and that will complicate clinical study design as well as assessment of the outcomes. So I think biomedical engineers could play a bigger role communicating with basic science as well as regulatory bodies.
Yeah, I think those are great points. I have to agree. I think, you know, I think as bioengineers, we're in a unique place where we can hopefully talk with both the clinicians and who are making the measurements and with the engineers who are sort of on the technology development side. So I think it's an exciting place to be. So I wanted to open up the floor for questions from the audience, especially from speakers. I mean, especially from students. I know there's a lot of students on this call, so we'd love to get some questions from students. Please just unmute yourself and go for it, students or faculty.
Kelly, if I could, I'd like to ask a quick question.
Sure, go ahead, go for it.
This is Lonnie. So to the panel, I guess one question that I have, I'm curious what the panel thinks is, so I very much like the precision health concept, but I'm wondering if we're measuring the right things in order to make the decisions. And I think we're often measuring things that we know how to measure, but is that really the things that are going to be needed in order for making the right decisions?
So, means if no one is starting, I can start. Like I think Alani had brought a very good point. So for an example, like we rely much of the mutational information from the patients to develop a target or uncover like new targets. But then in the clinic, not every patient is RNA-seq information is available, or even the omics information is available. So I don't know when that will happen. The insurance and the other issues are there. So.
And just to clarify, for example, it might be like,
yeah, so that's I
was gonna say, like, sequencing is a very common tool, but that's can be DNA level, it could be RNA, but maybe the protein level information is needed, or maybe you need to know the metabolites to actually know these other things. And so that's my question, like, how do we know which one of these things? There's so many things you could possibly measure. How do we figure out what are the right ones that are gonna be most predictive?
I guess, I mean, there are probably two answers. One, from a practical perspective, you know, if you find a marker or a test that gives you 100% accuracy in separating out different types of cancer, and that tells you, you know, this person's going to survive longer or shorter, I think that might be satisfactory from a practical point of view, but I don't know if that will capture the entire mechanism. You know, like maybe there are changes happening at the protein or metabolite levels that we are not measuring. So I guess there are the engineering side, if you're happy with an accurate, you know, like measurement that gives you a good prognosis, then I guess you can answer that easily. But the other part is probably the harder one. Do you actually have a big picture, complete picture of the disease? And I think that was going back to my question for James too earlier, you know, like from hundreds of genes, how do you pick the right one to, you know, like create an antigen for? So if something works really well, then I guess it doesn't matter.
Yeah, I think that's such an interesting comment, Lonnie. And I think it sort of goes back to like what we were talking about earlier with making, you know, the connection between function and molecule, and, you know, the connection between physiological scales and the connection between signatures and mechanism. And I wonder if it's, you know, we have all these technologies where we can make cost-effective, high throughput measurements, you know, at the molecular level. But I wonder if we'll sort of make improvements in the direction you're talking about if as we develop more technologies, make functional measurements. So, you know, I think, like you said, it's easy to get lots of data on molecules and proteins right now. It's harder to get functional measurements. And so I wonder if, as we develop technologies in that direction, maybe we'll be able to take steps that way. we have a couple more minutes if anybody else has a question for our panel or anything related to the student presentations or precision medicine. If there are, I guess, one thing that I'd be interested in hearing from the panel on is, where are we going? What's our dream right now? What can precision health do in modern medicine? In 20 years from now, what will be different?
Well, I feel like, each person's, I think one clear example for the microbiome, like your lab studying and James' lab studying too, is each person's microbiome is so unique. I think altering that for, improving health, living forever, like Carlos said, those probably are, I think, some of the commonplace technologies I would see, like maybe probiotic that says, hey, this will reduce your chance of cancer or improve your stem cells. So I feel like that could be a clear, precision, personalized microbiome therapy. Might be an easy thing I can see happening in a few years with so many companies already out there.
Just adding to Sriram, I think other than the microbiome and I think the diet, I think is also another angle, which is an upcoming issue. So relating all these parameters to diet will be another, I think we'll have a lot of innovations in that area, and that could become an important parameter.
Yeah, I agree. Personalized diet and microbiome, probably.
And I know Michigan Bio Initiative is putting a lot of resources on MIDAS and data science, and they are building a big consortium to apply AI predicting patient outcomes, and I think that would be very exciting to see in the next 5 to 10 years.
All right, thank you guys. Yeah, I think that's exciting to think about. And I guess if there's no more questions, there's no questions from the audience or students, we will conclude our panel discussion. And thanks
Sorry. I actually just came up with a question. Sorry to kind of cut in so late. First things first, I have to thank all the panelists for their amazing talk. It was really fascinating to hear all of you guys' facets of research. I did have a question regarding translating precision health to in a high throughput manner. I know that we touched up upon that near the end of the end of Professor Nargrath's discussion, but I was curious about how we actually translate precision health in a high throughput manner. To me, it seems like if you're trying to, I guess, base personalized medicine it seems like that's a very intensive procedure, right? Because you have to take into account an individual, an individual's I guess, biology. There's just so much that goes on in there, right? And that, to me, doesn't seem like you're able to, I guess, that seems to me you're not able to really apply that to every individual, kind of like how a high throughput manner is able to take in numerous amount of cells and easily screen them for such things, right? So I was curious about how we actually make that transition from precision health into a high throughput manner. And that's actually somewhat relevant to both Professors Chandrasekaran's and Professor Moon's research, right? Because in your research, you're looking at applying mechanistic AIs to determine the personalized therapy, as well as in Professor Moon's, you're looking at engineering well, this is actually not really relevant to his engineering nanomaterials, but more into his developing vaccine platforms, right? Where that can be used for developing personalized vaccines for like, how do you actually do that on a high throughput scale? I know that this may be a little bit jumping around, but yeah.
Yeah, well, I can maybe start. So here in Michigan, in Cancer Center, when patients are admitted, they are oftentimes sequenced for their DNA. So we could implement those neoantigen screening tools into the pipeline. But typically when they are screened for DNA, if you find specific mutated proteins, the patients are placed on clinical trials with that specific drug targeting molecules. So they are in clinical trials in California. They would be placed in trials using that drug in California rather than in Michigan and Ann Arbor. So these molecular tumor boards are in practice in Michigan Cancer Center. So I think that's a good example. And I think we'll see more of that across the nation in the coming years.
Yeah, I agree. I think many of the technologies already in place, like sequencing for James' case, and even for infectious disease like tuberculosis. Most hospitals, even in Asia or Africa, they have, like imaging equipment that they can use to measure chest X-rays or CT scans, or they could have like simple antibiotic susceptibility tests. So many of them are becoming cheaper, thanks to biomedical engineering. So most of the technology is already in place now. So you can, if you develop a right algorithm that can diagnose a certain disease or recommend treatments, I think things are already in place, and most of the algorithms could be run on a smartphone, and so I don't think those would be a limitation. So I think you will see this being widely used. And
I see, like using all that information, right? Then you can apply that algorithm for like various individual personal therapies. Okay.
I'll just add, and I think maybe some of the other panelists will agree with me, but I think computational approaches have so much potential for that, exactly that you're asking about, just because it's a really cost-effective way to quickly test hypotheses, in heterogeneous populations.
Yeah, exactly. Like,
yeah,
it's hell, I don't want to be on record saying this, but it's somewhat impractical to try to like constantly develop new therapies for one individual, right? It's much more practical to have it be almost applicable for everyone else, right? Because then you don't have to do as much work. But thanks for all your answers.
Thank you. Thanks for those questions. And thanks for our panelists. Thanks to our panelists and all our student speakers. And we'll go ahead and wrap up and move to the poster session, which will be in spatial chat. So thank you for being here, and look forward to seeing you next year.
Thank you. Bye.
All right, thank you. Okay, bye-bye.